Connaissance,
recherche,
sciences,
et sciences mathématiques,
théorie,
physique,
philosophie,
conférence, archéologie,
nature.
Right, yeah, so good afternoon, everybody, and thanks for having this symposium. I have a lot of very interesting talks, and the result of all of these interesting talks is that my talk keeps evolving rapidly. But what I did is then basically, let's see, we don't have a signal yet. Sorry?
Double display on your Mac.
Right, well, yeah, I guess I have to get something on the screen first.
This is a picture of the inside of the sauropod.
Right. Yes, right. Well, let's see if we get... Okay, well, it seems like we're up and running here. Right. So you see this title here is very, very general, and it turns out that that was actually a pretty good idea, and tells you very little about what I'm actually talking about. So what I'm talking about is something that really goes back to the first, or one of the things that Armand was famous, or became famous about, the question of dinosaur warm-bloodedness. And of course, Andrew Lee has just been talking about the same thing. But I would like to reflect as a starting point a little bit more, well, why should we be interested in this question? I don't think because the media are interested in this, but because it really is a fundamental property of an animal, and we need to know whether dinosaurs were warm-blooded or not to understand their evolution. And so, for example, there is a lot of evidence that you cannot be a very large animal, say above 1 ton body mass, without being warm-blooded. And that's sort of why, in my career, I've always shied away from addressing the question whether dinosaurs were warm-blooded or not. But lately, with our research, we're just really getting to the point where we are addressing this, and where personally I've realized that we have to understand this to understand body size evolution. So, well, and you see this has little to do on the outset of what my title says. So what my talk's really going to be about is two things. First, studying sauropods, I have had the interesting and unfortunate situation that sauropods grew so quickly that they are not laying down growth marks. And I've tried to turn this into a positive effect by erecting ontogenetic histologic stages so we can compare ontogenies. And that's, of course, the other thing that Armand has been very, very influential in, is that what we're dealing with in bone histology is fossilized ontogenies. And so for me, one of my highlights in my career was to be invited to a meeting on evolution and development. And so we can use absolute growth rates, we can use skeletochronology, but in cases we don't have this, then we should do something that embryologists do, which is have stages to compare ontogenies. So that's the first part of my talk. The second part of my talk then is trying to take growth rates and growth rate analysis and fitting of growth curves to as far as we possibly can. So let's get, as you know, I've been interested in sauropod dinosaurs, and again, not because they're of interest to the media, but because they're a real challenge to any evolutionary biologist, simply because they represent the upper limit of what can walk around on this earth, apparently, what has evolved so far. So that's why we need to study sauropod dinosaurs, and for me, personally, this started with the realization that sauropod dinosaurs are clearly so much bigger than anybody else. And you have here the usual mammals, your ornithischian dinosaurs, and then you have sauropods that always get bigger, and by an order of magnitude. And of course, so it's obvious to ask, well, what can we say about life history in these animals, and does it have something to do? So it's just as an introductory aside, really. So sauropod dinosaurs are big. And this then led to some technical problems. Of course, we always heard this throughout the symposium. We commonly have the problem of getting at the samples. So what we've done to solve this problem is it's difficult to cut across a sauropod limb bone that has this diameter. So we've taken a technique that's used in all kinds of fields, in dendrochronology, in medicine, where it's called biopsy. So we do biopsies of dinosaur bones using a diamond-studded core drill, and this is what you can see here. Of course, everything is a little larger than in medicine. Here we have this core, and here we have the drill. It's a normal electrical household drill, and we use that because then, just as in biopsy, it's extremely crucial to always sample at the same location. And of course, we would rather have the whole cross-section of the long bone, but again, there are technical problems. And because this is quick and easy, it also means that it will give us the large sample sizes where we can study ontogeny. So we don't only study one sauropod long bone, but we have, I don't know, about 250 or 300 different individuals through this technique with relatively little effort. And so, as I was saying, if you take sauropod long bones, have the advantage of having a very simple morphology, and so it's fairly easy to predict where to find the best growth record, simply because, they grow fairly evenly at both ends and in the circumference. And this really has been borne out by our sampling, so that we get away pretty much with this biopsy approach. And let's see. Right, and then the result is, that's where it becomes traditional. You basically, the core is a segment of the entire cross-section. It then gets cut into a thin section and into a polished section, and we study both. And even in the largest sauropod dinosaur, it's a fairly small section that is easy to cut. And then, this is where we are at with, Christy Curry talked about some sauropod dinosaurs. So this is a sauropod phylogeny, and all the red taxa is what we've sampled so far. So we're getting a pretty good coverage and really get samples from all over the place, and are able to make interesting comparisons. And so the vexing thing when I started out doing this is really that most of the time you really don't get a good growth record in these large long bones. So usually what you see is you see laminar fibrolamellar bone up to 6 centimeters of thickness without a single growth mark. So you're kind of stuck, and for a while there, I was unhappy about this, and then decided, well, to really look at this in more detail, and I realized there are actually subtle differences, and then sometimes they're not so subtle, that you have a basic correlation between the size of the bone and its histology, if you can be reasonably sure you have a single taxon. You have a growth series of a single taxon. And this then led to the idea of transferring the ontogenetic staging approach from embryos basically to the postnatal ontogeny of dinosaurs, and then using sauropods as an example. And so then we start out, I'm showing this to you in this one example here for basically basal neosauropods, diplodocoids, and basal macronarians, sort of your run-of-the-mill late Jurassic sauropods, Diplodocus, Apatosaurus, Camarasaurus, Brachiosaurus. And it works for all of these. And so we start out with a juvenile Camarasaurus, humerus, about this big. We look at its bone histology in some more detail. And then here for this, this is all published in detail, and so you can hopefully use this. That basically we have a pretty loose bone structure here. We have a laminar organization already, but we have large vascular spaces, and we're looking in detail at the amount of woven bone at the development of the primary osteons and so on and so forth. And for time constraints, I won't get into this in much detail. So here we have a Barosaurus humerus that's a little bit bigger. And then what we see is an increased laminar organization, but we still see clearly, still see clearly large vascular spaces. Of course, I also should say all of these are taken from the outer cortex close towards the bone surface because that represents the growth stage of the animal. And then an even larger one, that's pretty much the maximum that these diplodicoids get to reach. And you see still the laminar organization, typical of this type of bone, but the vascular spaces here are very narrow now. That means the primary osteons are fully mature and almost close up the vascular spaces. So this then we call stage 9. Again, I don't have the time to get into the details. And then in late stage ontogeny, if we have a very large Camarasaurus with a humerus about this high, then we start seeing growth marks in the femora. So, and then you see here there starts still the laminar organization, and then here some lines of arrested growth start coming in, and then eventually an external fundamental system, so we know the animal is fully grown. And this is basically then, of course, the end of its growth record proper, but it's not the end of its life history record, because you also see remodeling coming in, and these simple bones then happens in a regular fashion, so that now, if we look at if these fully grown animals, we can distinguish further stages based on the patterns of remodeling. And we're up to stage 14 now. So this is stage 13, where basically the entire primary cortex consists of reversion bone. That's then usually, and what we don't know yet is how much time is really in this remodeling. That's really the problem. Once the animal stops growing, then we have some growth marks in the external fundamental system, and so we can say, well, there were a few years of its lives that are still recorded, but eventually that will stop, or as many of you know, the growth marks in the external fundamental system get so closely spaced that you cannot resolve them in good conscience anymore. And so it would be nice to know what the rate is of this remodeling to then to characterize the post-growth ontogeny of the animal. But we can't really do that yet, so we just have these relative stages. And of course, all of this is without an absolute time calibration. The only thing we can say, of course, is that depending on tissue type, growth rate goes down until growth stops of the whole animal, but remodeling continues. And then this is then basically, of course, an expression of Amprino's rule, which I do not need to introduce to this audience. And then what you can do, of course, you can look at body size standardized to femur length. So we can use also, mainly we've used femora and humeri, and the humeri are calculated to femur length, and then compare this to histologic ontogenetic stage. And if we are in fact dealing with a growth series, then we should see a correlation between femur length and histologic, between body size and histologic ontogenetic stage, which is what we see in this Apatosaurus sample here. And so this is not terribly surprising. What we've not done, what could be done is, of course, you could characterize this correlation using a rank correlation, but here it's pretty clear that we haven't done it yet. It's not. And so, of course, then their example, here is Brachiosaurus, you see the same thing. Good correlation between body size and histologic stage, or histology, which is, of course, always the very basic thing we ask when we look at bone histology. And we've seen this throughout the symposium. Is there a relationship between the body size and the histology of the animal? And of course, that's some of my questions here during the discussions have been about size. And of course, when you study sauropods, then size is the overriding thing. And a sauropod grows from a baby to an adult through 5 orders of magnitude. So that drives home the importance of size. And then you see things like this, where you start losing this correlation. And here we just Camarasaurus is known to occur in different sized species in the Morrison Formation of North America, and this is the probable explanation why the correlation here is not very good anymore. And we've not tracked it down to individual species, because commonly we have long bones, and in the long bones we don't have the apomorphies of this specific species, but we weren't really interested in doing this either. It's just that the poor correlation correlates with the observation that they're different sized, or the claim or the observation by morphologists that there are different sized species in the Morrison. So, and that's what this reflects, that you have, for example, at a femur length, at ontogenetic stage 6, you can have animals that are obviously very small, and others that are much larger. But then what you can also do, and this is what the next talk, or the last talk here before Armand's talk will be about, is sometimes you get a clear separation. So here you have the Camarasaurus sample, and then we have this animal called Europasaurus, which is an island dwarf sauropod from Germany, and here's its growth trajectory, and it's well outside that of Camarasaurus. So, and that's the next talk will be about island dwarfing in these sauropods. And again, just because they're so big, it's so easy to see this kind of thing. So, and this is, for example, Brachiosaurus compared with Europasaurus. And so the very basic question you've always asked, well, is this little one a juvenile, or what is it? And so your null hypothesis would be that it's a juvenile. And then the second part of my talk, which pretty closely ties in with what Andrew just had to say, occasionally we get growth mark records, and then they tell us interesting things, and in this regard it also ties into, that's where then the thermometabolism became important. So in this case we're looking at Mamenchisaurus, where in ulna we have a very good growth record. It's a single individual, as you'll see, and the next here, Mamenchisaurus, one of these fairly basal, very long-necked sauropods, and a very large Mamenchisaurus ulna. This is an ulna that's 1 meter long, so that's a pretty substantial animal. This animal was then what one of my co-authors observed to have a good growth mark record. And here we actually did take an entire cross-section. There's not that much more left of the animal, but it's clear it was a very large sauropod. And then you can look at this growth mark in detail, and this growth mark record in detail, and then try to translate this into body mass increase, which is what Andrew Lee has done as well. And so just to show you how these growth marks are developed, they're here drawn in. But in the close-up, these are sort of the normal growth cycles here. They're not marked off by lines of arrested growth, but they're more of the type that Armand has called modulations, where you just see cyclical changes in vascularity. And then you get into the outer part of the bone, and you get an external fundamental system that has 6 growth cycles here in this case. And then you can plot, of course, cortex thickness versus age, and then we always have the problem that something of the growth record is missing, which, especially if you do a growth curve from a single individual, presents a particular problem. But you see the cortex thickness increase here, then here is the EFS. It's fairly regular, and this then you can also just look at the cortex thickness increase. Here as cycle thickness decreases, and here you have the EFS. But then that's what, of course, what a number of people have attempted. First, I think Greg Erickson, and then Andrew Lee and Tom Lehman, fitting growth models to these data. Here we've done something. We've gone from cortex thickness increase to body mass increase. And this, of course, also works in these fairly simple bones, where you have a pretty close relationship between the body size increase and the increase in thickness of the bone, or the other way around. And one of my co-authors then, who's good with these growth models, because she's an ecologist and they use this a lot, then has tried to fit different, very sophisticated models. Here a logistic model, and more commonly is the von Bertalanffy model, has been used to these data. And this is a von Bertalanffy model, and it assumed already a starting that 10 years maximum were missing. You see the fit with the data here, this is the model, this is the data, is not very good. But the logistic model then actually gives you a very good fit, and the nice thing about it is, as some other people have shown as well, that it predicts the number of missing cycles, which is actually only 3 here, and then you can do model selection and decide what you like best. So we like this model best, and as Andrew is pointing out, then here you can read off maximum growth rate. And maximum growth rate is something that, of course, has been, there's sexual maturity, generally assumed to be at the inflection point of these curves. And then, of course, you can look at the maximum growth rate from the observed cycles, but also from the model cycles. And then here you have a peak growth rate in kilograms per year. You transform this into growth rates. I'll go on to the next one here. Right, into growth rates and maximum growth rate in grams per day. And so what you can do with this then is you can actually test a recent hypothesis published by Brian McNabb, who just said, what about if sauropod dinosaurs only had 1/5 of a metabolic rate of a mammal, like in a varanid, then they would have used less energy and could have grown to a larger body size. So basically body mass, if energy increase is energy demand here, field energy expenditure is less, then the individual could grow larger. But this only works if you have low growth rates. So then you basically you can test this hypothesis by using what I've called Case curve. Case did something very interesting, and that he related maximum growth rate in grams per day with adult body mass. So there's a scaling in there, and that's why I always asked about body size. And he came up with these regressions that are more or less well supported, and noticed that reptiles and fish actually have much lower maximum growth rates at the same full body size. And then this way we were able to test McNabb's hypothesis, because here you have this 10-fold increase in maximum growth rate is correlated with a 10-fold increase in basal metabolic rate in mammals versus reptiles. And then what we did is we compiled data for varanids, because it was explicitly McNabb's hypothesis that varanids have 20% of the basal metabolic rate of a mammal, and so we did a case curve for varanids then. And then what you do is, here's the case curve for varanids, and then you use the dinosaur points. In this case, Mamenchisaurus. We could have plotted some others, but we didn't feel they were well enough constrained to do this. And we see that Mamenchisaurus still falls well above the varanid curve. So we're still in the process of working on this. And then this seems to falsify McNabb's suggestion that sauropod dinosaurs were basically overgrown varanid lizards because they are growing too quickly. And there we had an encounter with the metabolic theory of ecology, because one of the editor at PNAS, where we had sent this to, wrote, "I am convinced that Mamenchisaurus had a very high growth rate. The question is, does this proof imply that it also had a very high basal metabolic rate?" And what George Kubo has shown us, that yes, this seems to be the case. And then the second reviewer just said flat out he doesn't believe that there is this link between maximum growth rate and high basal metabolic rate. And so where we're at now, and I welcome suggestions, is of course, how can we nail down this relationship between high growth rates and high basal metabolic rates? And I think everybody would agree that there is a relationship between maximum growth rate and basal metabolic rate. And so this is just what I want to share with you. So in general, sauropod dinosaurs have this laminar fibrolamellar bone with rare growth marks, but the qualitative growth record can be captured in histologic ontogenetic stages, and I think that's something that can be done in various other animals. So it allows taxonomic and evolutionary insight, and the rare quantitative growth record then leads us fairly quickly to the issue of dinosaur basal metabolic rate. Thank you.